very good morning, everyone. I hope I am audible. Can anyone confirm it in the chat box? Okay, great. Manoj, what Manoj is saying yes. Okay, great. So, in yesterday sessions, what we have done, we have discussed about what actually I hope every one of you get an ideas what actually the network pharmacology is, what is the difference between system biology, network pharmacology, and quantitative system pharmacology, and PBPK modelings, and where you can apply these techniques. We have already covered in the last sessions graph theories. You get to know about what are the nodes and edges when we talk about network, simple network. What are the different types of graph? We have already discussed steps in network pharmacology. We have I just explained you the first step. So today's session is about how, if any one of you want to do network pharmacology for your research, so what are the steps, what steps you have to do. So that we are going to cover today's how to create the files, how you can identify the common target, how you can check the interactions, and of course analysis is very important part. So so how to do the analysis of the network, what are the different parameters you have to be so check for, and how to identify the pathways. So this already we have covered, so I am skipping this part. System biology, what are the different components under system biology, what skill is required when we talk about if you want to become a good network scientist. These are the simple equations, quantitative system pharmacology. Then we have discussed just a simple understandings about how these things we are doing, and then quantitative system pharmacology versus PBPK models, single drug, single target concept. We have discussed it in details. What is single drug and single target concept, and network pharmacology, how it helps pharmacology. So of course every one of you knows about what is pharmacology, and I told you before using these network pharmacological techniques. For example, someone of you is working in diabetes, so you should know about intracellular signaling pathways which are involved in diabetes. So before pathogenesis, you should understand how glucose level is maintained in our bodies, what is the mechanism behind it, what are the possible signaling by which insulin maintain our blood glucose levels. So all these signaling first you have to be understands, then you have to be understands what happens that blood glucose level is not maintained, what intracellular signaling is altered, and then so you can think of the drugs, and then you can better interpret the results of network pharmacology. This already we have covered, starting from the social networks, then these networks, biological network. We have already covered single target to multiple target. What are the importance where you can apply? We have already discussed in detail in last sessions. Popularities, I also maybe someone of you already checked in the PubMed. Just add the word network pharmacology, you will find that how popular these techniques are. Important signaling, as I already told you many times, go through the normal intracellular signaling pathway. Of course, for whatever the disease you are working, you should know about in detail about intracellular signaling pathway of that particular disease. But apart from that, there are some signaling pathways which are involved in multiple disease. Like if we talk about apoptotic signaling pathways, is involved in various types of disease. When we talk about immune system is altered in various disease, not in single disease. So all these pathways, JAK-STAT signaling pathway we talk about PI3-AKT. You just do the Google, you will find that PI3-AKT is the signaling pathways which are involved in multiple disease, right? So that mTOR signaling pathways, and you should understand about. And I told you yesterday as well, try to correlate your findings with the clinical data as well. This already I have explained you. Simple network history, Conisberg bridge problem, you can go through it. That is the Euler mathematicians from where graph theory has been merged. And graph theory is the ultimate study the relationship. So these already we have covered. What are the different types of graph: undirected, directed, weighted, unweighted, cyclic, acyclic. Cyclic, acyclic is further can be directed acyclic, undirected acyclic, or cyclic directed, or cyclic undirected. Then we talk about sparse or dense graph, but of course in biology or pharmacology, we are dealing with the dense graph, not with the sparse graph. Then different types of network. Someone of you also asked yesterday that can we go with the protein network? Yes, you can go with protein networks. You can check that part as well. Then these are the small world network, scale free network, modular network. So when we talk about if you want to understand pathogenesis of disease, it can be small world network, it can be modular network, it can be random as well. But when we talk about drugs and their possible mechanism in particular disease, we mainly dealing with the bipartite network. So different types of networks are there: gene regulation network, protein network, signal transduction network, metabolic network, and so on. So now today's session is on the steps which you have to do. So I hope every one of you is with your laptop or with your computer systems, so that step by step you can use these web servers and pull out the data in the Excel file. And I told you yesterday as well, first two steps are very important with respect to network pharmacology, because that are the foundations for any network. After these two steps, next steps are, particularly you are playing with different tools, you are creating the network, analyzing the network. But the root is, or foundation is your these two steps. And what are these two steps? So you should give much more time to these two steps. Never be in hurry for these two steps. So, because these two steps are time consuming process, right? It will take time. You will, first thing is you should understand about the web server or the database from where you are pulling the data. So I told you first step is you pull out the targets which are involved in particular disease. As for example, you are working on diabetes, or someone of you working on Alzheimer, or someone of you working on arthritis, whatever your research area of interest. So first step what you have to do is, if someone is working on diabetes, they have to pull out all the targets which are involved in the diabetes. Then also you have to define which type of diabetes you are talking about: type 1 diabetes, type 2 diabetes, gestational diabetes. So you have to specify your disease. And by using these, whatever you seen in on your screen, you can take the screenshot of that one as well. These are the online freely available databases where you can find the informations about the targets involved in the disease. So this is the first step which you have to do. Are you getting my point? So this is the open target platform is the one. You just type in a Google. I can show you if you are with your laptop. So simultaneously you can also do these steps. So let me show you first one: open targets platform, right? I hope my Google is visible. Open target. Just type in a Google open target platform. Put in the chat box if you are doing it. Gaware Manoj, are you doing it? Sneha, Hafiz Cam 1, are you doing it? If you are with systems, okay, Isa Garg is doing. So just type in a Google's open target platform. So it will open like this, right? Just click on you, it will open like this, right? So for examples, I am adding here any disease name, non-alcoholic fatty liver disease I am adding, right? So I just add in the search. You can add in the search uh, particular disease in which particular disease or area of your interest, whatever. Gaurav, Gaurav is also saying he is also doing. Yeah, Gaurav, Isa, Hafiz, Sneha, Gaware Manoj. You can just type in a Google every one of you, open target platform, click on it, add the name of the disease, and click on search. So now you can see that I just added non-alcoholic fatty liver disease. So in non-alcoholic, I want to see what are the proteins, what are the targets which are involved in the. We are talking about, still we are not talking about drugs. I told you, first thing is we have to understand about the pathogenesis of the disease. First we have to identify the targets, then we can identify our drug where it can be target, right? So I just added the disease name. Now you can see that first one, PNPLA3 is the first targets, right? And now you can see that this open target platform is getting that data from all these. You can see that European PMC from literature, it's also getting the data, Reactome, Project Score, CRISPR screen, cancer biomarker, Cliniv, InnoGen, cancer gene census, ClinGen, Orphanet, Uniprot, right? Gene, ClinVar, Gene Verdon, Gene Wide Association Studies data. So now you can see that this target PNPLA3, you are getting the association score. Here you can check the association score is there. It's a cumulative score of all the available resources. So score is 0.74, right? So zero point 0.7 score means there is a great involvement of this target in the development of NAFLD or in the progression of NAFLD as well. You can see that here association score. There is a 0.02 1. 0 to 1 is the scale. So if you are close to 1, that means there is a strong associations. If now you can, if you go to the next page, then next page you will find that their association score will be less. This is a PDE 6G, but you can see the score is 0.31. So 0.31 is falling this side, so it's less than 0.5. So that means this target is not playing much more important role in the development. It's a role, but it's not a means still now you don't have a very strong evidence for it, right? So you have to check for the association score. You can see this, right? Association score. You can search specific target as well, but you can look for the association score here, and you can pin target, you can check target interaction, navigate to the profile, navigate to the evidence, navigate to the association as well. If I navigate to the evidence, so I will get to know that okay, from where this informations we have, so there is a gene wide associations platform where that target has been identified. There is an article on it, Europe PMC. So this article is highlighting the role of these targets in NAFLD. If you are working for any other disease, so you can see that part as well, right? So you can this open target platform is very important platform where you can get targets with association score. So sometimes we are not interested for such kind of target whose association score is very less, right? So any question so far? Subham saying good morning. Good morning. Hamish Darshan 7104. Okay, yes sir. Okay. So you all are doing it. So you can export the data as well. You can download the data. You can upload target as well here. So what I mean to say is, whenever you are using any platform, whatever platform today I am showing you, first thing is, important thing is, read about that platform. Like, if you want to use open target platform, do not use it blindly. First read about open target platform. You can see that there is a documentation is there, that you will get to know about from where this open target platform is getting the data, what should be the cutoff values, what is the target disease evidence, what is the target disease association, what is GWS and functional genomics. Are you getting my point? There is a FAQ as well. In FAQ you will get the common queries which arise in everyone's minds. So that is also, you will get information from here, where you can read about what are the quality control criteria for fine mapping, how they have mapped the data, from where they are getting the data, Then according to the your research questions, you can think of whether this database is important for me or it's not important for me. As I said, if you are, for example, working on orphan disease, right, rare disease, so by reading about this database, you get to know that no, this database does not contain these particular disease data. So what is the point to include that? That's why I'm saying selection of database should not be done blindly. Selection of, first thing is, you should know about what are the different databases from where I can get the informations about the targets which are involved in the disease. Then read about that database, go to their FAQ, go to their documentations, read about their notes, which is available on their website, and then you decide whether for my research is this database is important or not. Are you getting my point? So sir, you should understand about what are the quality control criteria for fine mapping. What is V2G score, which is not available now, they have converted into another scores. What should be the cutoff, right? Data code and access, you can get more informations in bibliography as well, what how they have done. So sir, you have to do NER as well, but they have also done the NER. They have done entity normalizations, literature based similar entities, open AI literature summarizing tool. And then you will get sir, these publication as well, where you will get to know much more detail about this. Are you getting my point? Any question on this? So what I mean to say, whenever you are using any database, read about it. These are very powerful database, you, but problem is, if you don't know how to use in a optimum, means in a right way, means you will always will be in trouble. If someone asks you query, okay, in open target platform, what is the basis of cutoff values, how these cutoff values are decided, and why you have considered that, right? So you can take screenshot of this. These are all are the databases which you have to be pull out the data regarding targets involved in disease. DigiNet is another one. DigiNet nowadays is asking for your institutional ID. So if you have an institutional ID, you can do in the login in the DigiNet and add in the search about the disease, and you can pull out the targets. Like there is a gene card, OMIM, HPO, Mala card, Orphanet. Orphanet is particularly for the orphan drug. Just one second. GWAS. GWAS, that is a Gene Wide Association Catalog, Hughes Navigator, ClinGen, that is a clinical genome resource, and of course, also you have to do text mining approaches. So this is the first step for the network. If you are planning for a network pharmacological studies, this is the first step where you have to, understands about the disease, and to understand about the disease, you have to pull out data from all these databases. And apart from these databases, if you are getting by reading about these network pharmacological papers, research studies of good journal, if you are finding that they have included one more database, include it. As I told you, this is the area where you have to be update yourself. So maybe one database is not there, or two new databases you come across, so include it, right? So this is the first step which you have to do. Then second step is identification of targets of compounds. So of course, when we are talking about the pharmacology, we are talking about the drugs, new chemical entities. So there are two files which you have to be create. One file is will contain the target which are involved in disease. One more Excel file which you have to prepare, which contain all the targets which your compound can hit. Like if someone of you working on natural compound, so you should have the structure of that natural compound, and then in these databases put the structure or smiley notation of that particular compound, and pull out the target informations in Excel file, right? So these are the two very important steps when we talk about network pharmacological techniques, and give much more time to these two steps, because these are the foundation step, as many times I told you. because later on when you created the network, analyze the network, if any target is missing, so that means that target is not there in the foundation files. Next step is common targets. You have to identify the common target between compound and disease. Then you can check the interactions among the common targets by string database, biograde, deep stitch, inact are there. And then you can create and analyze the network by using the network x module of your Python and igraph module of R. You can use either one. And if still if you are not comfortable with Python or R, then you can go with the window based tool, that is a cytoscape. By that one also you can do it, but you can't do much more analysis by that. And for pathway you can do the SR plot. So now we are going to get into detail about first step which you all have to do: targets involved in disease. So as I said, target involved in disease, one of the databases DGNet. So in DGNet you can see that there is a option to sign up. So as I said, for DGNet you should have a institutional login ID and password. So for DGNet you just type in a Google DGNet. DGNet. DGNet, a very popular database if you want to understand about the targets which are involved in particular disease. So you can see that of course now it's become subscription based as well, but free trial you can get it. But before that you can create your login ID and passwords. It's This database contains huge number of data is there. So first thing is, as I said, before using any database, go through their documents, go through, understands about how this database has been created, what is its attributes, what is metrics, what is its association ontologies, and means basis of their from where they are getting the data. That is very important, right? So gene disease association, variant disease association, you can see about the data, how much data, these are the various releases of the DGNet, attribute, disease association type ontology. So all there is a schema is also there, so you can get to know about how this DGNet is formed, what is the schema. So you can see the flow diagram as well, schema of this one. You can keep it large and you can get the informations. So what I mean to say, whenever you are using any tool, you can see that there is a R package integration is also there, REST API, cytoscape is also there, AIDA enrichment is also there. So you can integrate their data informations with these all. About, you can read about it, what is DGNet, what is the basis, how it's created, everything, right? Then once you create your login ID and passwords for the free trial, earlier it was free, but now they have subscription based. But you can use the free trial as well. You can do the login. These are my login IDs in the DigiNet. And after that you can add in the search about the name of the disease, and just click on search. You will get like this summary of gene disease association. So when I click on summary of gene disease associations, you will get the data like this. Like I have added in the search drug resistance epilepsy. So you can see that these are the genes which are involved in the drug resistance epilepsy. And I am getting the gene ID, Uniprot IDs. So these are the genes which are involved in the drug resistance epilepsy. So I will pull these data in the Excel file. But if you are unable to assess the data of DGNet, means after using the free trial, you have done one project by using the DGNet, but for the next project you don't have, because DGNet is asking for the subscription. So there is a gene card is also there. So in gene card, you just in the search you can add the name of the disease. Like here we have added gram negative bacteria. So in gram negative bacteria, these are the targets which are involved. And of course when we talk about bacterial infections or fungal infections, you will get the target related to immune systems, not with respect to bacteria or fungus. Like in bacteria, if you want to, if your experts say why not there is a penicillin binding protein, why not there is a beta lactamase, why not there is a topoisomerase. The reason is because these databases is providing the informations about homo sapiens. So once your bacteria enter in the body, after that, so you will find these immune systems targets you will find in these databases. So that is one of the limitations for with respect to infectious disease. But with respect to other disease, you will get the targets, right? So gene card is another one. So just type in a Google gene card. Is anyone is doing it? Gene card. So you can see that gene card, the human gene database. So you can, again, first you read about gene card. Here is a option for mala card as well, path card as well, gene answer as well, gene analytics as well, gene like me as well. So unless and until you can see that there is a user guide is also there. For every databases there is a user guide, there is a all information is available how these databases are formed, and so what is the significance. So you can see that there is a mala cards, other cards are also there. So you can add, so you can explore. These are particular, for example, you want to see, okay, this is a particular particular disease, for example prostate cancer, click on that. These are the BRCA2 is the mainly gene which is responsible for this prostate cancer particularly. So you can click on it, you can check its association score, you can check source of information as well. So these databases contains data from multiple sources. These databases contains data from the experimental part as well, from the clinical part as well. So you can cross check what is the source from where you are getting this data, right? You can see that it's providing the relevance score. So you can get the informations. These are all are the second one, third one, fourth one. So you can see that their knowledge and their relevance score 138, then 113. So these are arranged in the order as per the relevance score. Relevance score means this BRAC C2 is has uh, uh, found in more number of articles as compared to other one, if it provides the data from the articles, right? Studies, more studies has been done which shows that BRAC2 is related to your I think we have added pancreatic cancers, right? So that's why your this relevance score is high. Then second one, androgen receptors, this one like that, right? So you have to decide, okay, and you have to read their cutoff values as well. Up to this relevance score I have to include. For example, you want to include only the top 10 targets or top 20 targets only, and you want to go with that one only, you want to check their interaction only, your research question is like that only. Sometimes you want to include all, then later on you can refine it, because there are a lot of number of these one databases are means targets are there. And here is your options to you can do the login, and after doing the login or registration in the gene card, you can download these targets in the excel file. So in excel file you have targets, means their symbol and their complete name, right? And you can put the source name. Okay, these are the targets from the gene card, these are the targets from HPO, these are the targets from the mala card. So like that you have to be create the files. This is a gene card. Another one is your man omim is this is a omim database. It's a comprehensive database on human gene and hereditary traits. So it's tell you about the relationship between disease phenotype and genotype. And major advantage is it's updated on daily basis, and database was initiated in early 1960, by the scientist. Catalog of Mendelian trait and disorder, Mendelian inheritance in man. The online version was created, so you can read about who has created it, how these databases has been created, what are their advantages, what are their limitations. First you understands all these things about the database, then only you decide whether for my research question this database is important or not important. you again I am telling, what mistakes we are doing is, you taken the screenshot. Okay, for disease these are the 10 databases where I have to pull out the data in the excel file, and without knowing about the database you always do the mistakes, right? You always do the mistakes. You don't understand about the cutoff values. What is the cutoff value for data, for that database? Where I am getting this data? This database is getting data from, what is the source of data for this database? That is also important to understand. As I said, some databases have a data, experimental data only. Some has clinical data. Some has data from the literature. Someone has data from the other sources. Someone have data from the all the sources. You getting my point? So you will get to know about after going through these. And this is the omim screenshot. Omim database, 50 years they have completed an online catalog of human gene and genetic disorder. So so if you are particularly working on genetic disorder, you can't miss out this database. And here is, you can see that omim video tutorial is also available. So if you type in Google omim, omim 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 database, you can get it. Anyone has any question, you can put in the chat box. And you can also do these steps. They are asking for the donations. It's freely available. So now you can see on the screen omim database and online, and you can see that when it's last updated. It's updated on 15 June 2026, right? 2 days back it's updated. So you can see that you can add here the name of the disease, click on search, you will get the informations about that disease, right? For example, any disease you can add. For example, I am just adding cancer. You can add particular cancer as well, right? I'm just adding any dot. Now you can see that these many targets which are involved in that particular cancers. You can download this and download as excel file. You can download all these targets in the excel file. But again, you have to be first, again and again I am telling to all of you, is first read about this omim, online mammalian inheritance in man. You can see that how this has created, what is the source, what is the statistics, what are the phenotype gene statistics associations. Download mi match is also there, so you can create your login ids. And before using this man omim database, you can see that there is a video tutorial is also available, right? Gene shoot, how to search tutorial, mim tutorial. So how to search in the omim database, you can get in the videos as well. So this is the omim, right? And if we talk about scoring method of omim, in omim you will not get numerical values, because omim is using qualitative evidence classification of gene phenotype relationship. So you will get signs like that symbol, it denotes phenotype with known molecular basis. This percentage indicate disorder with suspected Mendelian inheritance but without an identified causal gene. This star symbol indicate gene with known sequence informations, plus represent entries describing both a gene and phenotype together. So this type of qualitative based classifications you will get. That is a scoring method of omim database. Whereas gene card, you got to know about the relevance score, right? There is a quantitative numbers, but in omim there is no quantitative numbers. You will get like these qualitative evidence classifications. So so again, depending upon your research question, sometimes we want to include only where high relevance score or there is a high associations of that particular targets in particular disease. We want to consider only that one. So omim emphasize experimental validated causality rather than statistical association. So it does not provide a confidence score, p value, quantitative threshold. Already I told you. So there is another database, that is the HPO. HPO is a human phenotypic ontology database. It's created in 2007 in Charité University Hospital in Berlin. Provide information on medical related phenotype, and it's NIH supported projects, and it's available on this website. You can, if you add HPO, you will get in Google as well. HPO database. HPO. So you can see that human phenotype ontology. So again, read about this database. What is the basis? There is a documentation is available. Information is available. Guide is available. Annotation is available. Developer tool is available. So you can first read about this. What is the hierarchy? How this HPO is developed, and from where you are getting the data. Then you decide whether it is important for me or not, for according to my research questions. So this is the HPO. HPO is another one, human phenotype ontology. Then there is a Malacard. Malacard is another one. Malacard is human disease database, and it integrates both specialized and general disease list, including rare disease, genetic disease, complex disorders, and more. This is the screenshot of Malacard. So you can see that if you type in a Google Malacard, it's a human disease database. It's also very important database, and you can see that Malacard, Gene Card, Path Card, these all are integrated with each others. So you can see that in Malacard, you can see there is a. If you type in a Google Malacard, just type in a Google Malacard. So you can see that Malacard, comprehensive integrated human disease database, right? So I hope today's net connection is okay and everything going fine. Yeah, Gavare Manoj is asking which database is best among all these and how to choose the best one. So I already said, first thing is you should read about all the databases, read about their basis. Like some databases are for if you are working on so genetic disorder, so that databases are particularly designed for genetic disorder. There are some databases which are designed for orphan disease only, right? So you can't miss out that databases if you are working on that particular disease. If there is some database which specifically on orphan disease, and if you are not working on orphan disease, so there is no point to include that database. So that's why Gaverima knows what I am saying is, first read about the database. Documents are available on their websites. What is the database, from where they are getting the data, what is their scoring methods, what is the cutoff values, how cutoff values are decided. Then you will decide what is my research questions, whether this database is important or not, whether I have to keep the cutoff values or not, because that is depend upon your research questions, what you are looking for. If you are looking for a new targets or something new, so you can't exclude anything, so you have to include. And then of course analysis or network will become very complex, but you have to do that analysis. Is it clear, Gavare Manoj? And your question is which database is best among all these. All database has their own advantages and limitations, so I can't say this database is best, this database is not best, right? Okay, great. So Malacard, you can see that human disease database. So in Malacard, you can add the name of the disease, and you will get the target. So there are tools and databases as well. You can see that you can click on help, where you can get the search guide. You can get user guide as well, right? How to use and what is this card, what is overview, summary, associate genes, from where you are getting the data, what is the gene enrichment, and means geoterms they are using, what are the pathways they are using. So all information you will get. And when you have a good knowledge about the database, so so you can explain to the others in a better way. Yes, This is the advantage of this the particular mala card. And according to your research question, this is the best one for you. You have to be, you can't exclude it, right? So read about mala card, so you can see that there is a release notes, their publications are also there. You can see their publication as well. Mala card, the human disease, regional of gene and disease, NGS interpretation by gene card, mala card, Malakard. You can read about these publications are available, right? Complete list of our publications. So these are the publications where Malakard has been used, right? And in these publications, you will get to know about how these databases has been created and what its advantages and limitations, right? Any questions up to this? So this is the your Malakard. Then there is a another one is open target platform. Open target platform, it's a integrative bioinformatic resource developed to identify and prioritize therapeutic target associated with the human disease. So this platform combines large-scale genomic, transcriptomics, proteomic, pharmacological, clinical data set. So all these data sets combined together make one database. That database is open target platform, and it's established through collaborations among European Bioinformatics Institute, Wellcome Sanger Institute, GlaxoSmith, Biogen, with the aim of improving therapeutic target identification using integrated omics data. So omics we have talked about yesterday as well, right? The word omics, like when we talk about genomics, उसमें भी omics है, transcriptomics omics, metabolomics omics, proteomics omics, right? Epigenomics omics. So all these combined together we call omics data, right? So this is the open target platform I already shown you. And scoring methods of the open target platform is primary quantitative. It provides the scoring in the quantitative number, and that is the association score. Already I have shown you a normalized score ranging from 0 to 1, and that reflects the overall strength of the evidence linking a target to a disease. Ki how much this target is linked with that particular disease. If you are getting more association score, that means that target is more linked, strongly linked with that particular disease, right? So the score is generated through weighted integration of multiple evidences type like genetic associations. So this database contains the data from all these sources: transcriptomics, pathway, known drugs, somatic mutations, animal model, literature evidence. So based on that, it creates the score. Another thing is when we talk about open target platform is their early version is using so now they have a little bit advanced machine learning based locus to gene prediction model they are using, and this locus to gene framework represent a major advancement by improving the assignment of disease associated genomic loci to likely causal gene using fine mapping and functional genomic evidence. So you can get a more fine informations. So because now they are applying this L2G model, and this L2G model integrate all these mapped variant, disequilibration, chromatin integrations, distance to gene matrix, expression quantitative trait loci evidence using supervised machine learning approaches. Anyone of you knows about supervised or unsupervised machine learning? What is the difference between supervised and unsupervised? Can you put in the chat box? Because sir, when we talk about machine learning is, what does it mean according to layman? You are you means सबसे पहले तो आप मशीन को सिखाना पड़ेगा आपको. मशीन इज जस्ट लाइक ए चाइल्ड. पहले आपको बच्चे को सिखाना पड़ेगा कि भाई अगर तुम्हें रोड क्रॉस करना है तो तुम्हें इस साइड भी देखना है, इस साइड भी देखना है, सब क्लियर है तब तुम्हें रोड क्रॉस करना है. तो फर्स्ट यू हैव टू मेक him or her learn. So like that, you first do the machine learning. Machine learning means आपको मशीन को पहले सिखाना पड़ेगा. Are you getting my point? Then when your machine learn, then it will do the work for you, right? Like for example, similar patterns they have learned. Then there is a supervised machine learning approaches, unsupervised. So do you know, anyone of you knows the difference between supervised machine learning and unsupervised machine learning? Yeah, Gavere Manoj is saying machine learning involve artificial intelligence developing to machine. Yeah, but Gavere Manoj, machine जैसे फॉर एग्जांपल आपने कोई मशीन बनाई, पहले तो आपको उसको लर्न करवानी पड़ेगी वो चीज़, तभी वो काम करेगा, कोई भी चीज़। या नीतीश सिंह, known results, unknown results. Yeah, you are somewhat close to answer, because supervised means you have a labeled data, right? And in unsupervised means you have a unlabeled data. Data is not labeled, right? That you are putting in terms of known result or unknown results. Even AI agent walks off mixed puzzle places with a picture on the box to guide it. Yeah, it's your answer is little bit, you know, complicated, Gaurav. You can simplify it, but in simplification means supervised means you have a labeled data. Data is labeled, so you can learn your machine. This is the data. This is particular pattern is there in the data, and accordingly machine will learn, right? So what why I am saying this, because now is the era of AI, machine learning, and all some of these databases are using machine learning algorithm as well. So unless and until if you don't read about the database, and that is a major problem which I have observed, is students don't know about the basis of database, student don't know about the cutoff value, student don't know about who has created this database and what is the basis of that database. You simply what you are doing, आपको तीन चार database के नाम पता चल गए, आपने बस वहाँ से उसमें नाम डाला, वहाँ से एक्सेल फाइल अपलोड कर ली, उसको फिर वहाँ उसमें वैनी कर लिया, उसके बाद कॉमन टारगेट से स्ट्रिंग कर लिया और साइटोस्केप में नेटवर्क बना लिया, बस. That is not the best way to do that, right? If you are explaining to somewhere, so if you are adding the flavor of machine learning, supervised machine learning, unsupervised, this is a L2G model, and you are, because that database is providing data based on that, so why you are not able to explain it? You should read and explain it. And to use these, beauty of these databases is these are all are window based. No one is very complicated to use, very simple to use, right? Everyone can use these databases, right? So association score, when we talk about this, is above zero point 0.3 are generally considered moderate evidence. If it's between zero point 0.5 to zero point 0.7, it's a strong target disease associations, right? So what I mean to say, go through the databases, read about the cutoff. There is orphanet, another database is orphanet. Orphanet is, as its name indicate, it's an international rare disease knowledge base. So if you are working on a rare diseases, right? So you can't miss out this database. So you use this database. Orphanet was established in France in 1997, and you can see that you just type in a Google's, and scoring methods, it use qualitative evidence based on classification. And as I said, some databases are using scoring methods qualitatively, some are using quantitative number, so you should know about it. Some are using p value, some are using relevance score, some are using association score, some are using qualitative classification. Like in this one, you will find they are classifying as a established, probable, or limited based. So you should know about that. Okay, this is the limited evidence, this is the probable, this is the strong evidence, this is the particular database who is providing the information on that one. And according to my research question, I want to include only the targets which have a strong association with that particular disease, right? So you can see that disease are categorized according to clinical validity, prevalence, inheritance pattern, and molecular characterizations. There is another database is GWS catalog. GWS, I hope many of you might heard about GWS. This is a gene wide association studies. It investigating association between genetic variants and human trait or disease. It's launched in 2008 through collaborations with NHGRI, that is the National Human Genome Research Institute, and EMBL, that is the European Bioinformatics Institute. So this is the screenshot of GWS catalog. Just type in a Google GWS catalog. You will find it. Again, before using it, read about it. There is a options, download, learn. Click on that. You will get the materials about the GWS catalog, and then use it for your research. Scoring methods, when we talk about scoring methods of GWS catalog, it does not generate independent predictive score but stores statistical metrics reported in GWS studies. So it provides the data so based on the GWS studies which are published in the literature. GWS is gene wide association studies. So in these they are using p values, and p value measure the statistical significance of association between the genetic variant and trait. So these values are generally derived from regression based statistical model, including logistic regression for binary trait, linear regression for quantitative phenotype, and threshold is, you can see on your screen, 5 multiplied by 10 raised to a minus 8 to account for multiple hypothesis testing across the genome. Additional metrics include effect size estimate like such as beta coefficients, odd ratio, confidence interval, risk allele frequencies. So these terms you might heard when we are doing GWS studies. I hope many of you are doing gene wide association studies as well. So in gene wide association studies, this database is providing the data for us, right? And beta coefficient indicate magnitude and direction of genetic effect. Odd ratios quantify disease risk associated with specifically. Then another database is Hughes Navigator. This is another one. It developed by CDC, and CDC is Center for Disease Control and Prevention. That is a part of Human Genome Epidemiology Network initiative. It's a big network, right? And it providing the data based on biomedical literature mining indexing system rather than a predictive computer platform. It's This database is not doing the predictions, it's providing the data from the biomedical literature. So this is the, screenshot of this public health genomics and precision health knowledge base. You can see on that is a Hughes Navigator. So you can use Hughes Navigator. If we talk about scoring methods of Hughes Navigator, it does not employ a unified quantitative scoring framework. It relies on the evidence aggregation derived from the published literature. So database primarily reflect the number, frequency, and consistency of reported gene disease association across the studies, right? So there is no universally accepted cutoff when we talk about Hughes Navigator, right? Then there is a ClinGen. ClinGen is a clinical genome resource. It's a genomic knowledge database established to evaluate and standardize the clinical relevance of gene and genetic variants in human disease, and it employs a structured evidence-based scoring framework to assess the strength of gene-disease relationship, and it's launched in 2013 as a National Institute of Health funded initiative aimed at creating an authoritative central resource for clinical genomic interpretations. So this is a screenshot of ClinGen. Just type in Google ClinGen and read about ClinGen and use for your research. Scoring method of the ClinGen is it uses a semi-quantitative evidence scoring framework to classify gene disease validity. It classifies gene disease relationship into category including limited, moderate, strong, or definite. If you are getting score between 7 and 11, it indicate moderate evidence. If you are getting score above 12, it sports strong or definite classification when replicated over the time. Then there is another one, GenClip 3. It's again literature mining and functional enrichment platform designed to identify relationship among gene, biological process, pathways, and primarily using co-occurrence frequency and enrichment statistics to evaluate gene keyword relationship. And the platform commonly apply hypergeometric distributions. So what I mean to say, whatever the databases I am showing to you, read about what you have to read. Read about who has created it, from where you are getting the data, what is its advantages, what is its limitations, what is the scoring methods, what is the cutoff values, and what the cutoff value represent. So in this case, cutoff value is represented by the p value. Lower p value indicates stronger enrichment significance with thresholds such as p less than 0.05, right? FDR is a false discovery rate less than 0.05, commonly used to identify biological relevant association. So these cutoff values you have to write in your projects, in your thesis, in your research manuscript. Then your manuscript will become effective. Otherwise, what problems I am observing is the paper which are published with respect to network pharmacology, they are not putting much more detail. They are just writing. They have used this database. That's it. And even in some articles, researchers are using only one database. But one database is not enough, because unless and until if you are using any commercial database which providing all the comprehensive information, but when you are using freely available databases, you have to include as much you can. Now question might be arise in your mind, if we are doing same thing in a different databases, the targets may be overlap. There is a possibility or not? Can anyone answer this question? Yeah, Avdesh is saying right. Supervised machine learning provide correct, organized, and structured data. Yeah, labeled data, whereas unsupervised, less accurate, unorganized data. Right. Right. My question to all of you is, Abhi, whatever the databases I have shown you, this is a just let me complete this databases. Then I will come to the question. This Gen Clip 3, you just type in Google Gen Clip 3. It's open like this, and you can pull out the datas and cutoff values of CTD. Genes were selected using the inference score, cutoff values more than or equal to 100. Open target platform, genes were shortlisted based on global score. DG databases, gene with Z score. So what I mean to say, every make your notes, गो थ्रू ऑल द डाटाबेसेस और उनका एक टेबल बना लो. इस डाटाबेसेस में यहाँ यहाँ से इंफॉर्मेशन है, इस डाटाबेस में यहाँ से इंफॉर्मेशन है, इस डाटाबेस में यहाँ से है, इस डाटाबेस ये इसने क्रिएट किया इस ईयर में, ये तब अपडेट होता है. फिर उस टेबल में ये भी बना लो, इस डाटाबेस का ये एडवांटेज है, इसका ये लिमिटेशन है, इसका ये एडवांटेज है, इसका ये लिमिटेशन है. एक आपने प्रॉपर टेबल बना लिया, राइट. जस्ट गिव मी 1 सेकेंड. हेलो. हाँ. हेलो. हाँ हाँ. हेलो, बोलो भाई, बोलो. हाँ हाँ. एक बार कॉल कर ले उसको. ठीक है. तो पर्टिकुलरली व्हाट आई एम सजेस्टिंग टू ऑल ऑफ यू इज, आप एक टेबल बना लेना सभी डाटाबेसेस का. उसमें आप उसके एडवांटेजेस, लिमिटेशंस, उसके सोर्सेज क्या हैं, उसकी कट ऑफ वैल्यूज क्या हैं. देन दैट दैट पेज, एक बड़े पेज पे आप बना लोगे टेबल सब डाटाबेसेस का. when you are working on any research questions, you can decide कि ये मेरा comprehensive टेबल है सारा. मैंने एक चार्ट बना रखा है, चार्ट बना लो, क्योंकि बहुत सारे डाटाबेसेस है ना. दैट इज द फर्स्ट स्टेप. सो मैं अगर डाटाबेसेस के स्क्रीनशॉट पे जाऊँ, ये अभी डिजीज के डाटाबेस की बात कर रहे हैं हम, ये वाले. ये अब आप क्या करो, इसका ओपन प्लेटफॉर्म, एक कॉलम में डिजी नेट, जीन कार्ड, ओमिम, इस तरह आप सारा चार्ट बना लो. फिर इन डाटाबेसेस की वेबसाइट पे जाओ, इनका सोर्स देखो. पहला डालो सोर्स कि इस डाटाबेस में कहाँ से डाटा आ रहा है, डिजी नेट में कहाँ से आ रहा है, जीन कार्ड में कहाँ से आ रहा है, सोर्सेज ऑफ डाटा. तो उनके नाम लिख दो सब में, सोर्सेज, दीज आर द डिफरेंट सोर्सेज. फिर इनके स्कोरिंग मेथड्स, फिर आगे लिखो स्कोरिंग मेथड में कि ओपन प्लेटफॉर्म का ये स्कोरिंग मेथड है, डीजीनेट का ये है, जीन कार्ड का ये है, ओमिम का ये है, एचपीओ का ये है. आर यू गेटिंग माय पॉइंट? फिर आप क्या करो, उसके बाद लिखो एडवांटेजेस. तो जो स्पेसिफिक एडवांटेज है जो दूसरे में नहीं है, तो उसके नीचे एडवांटेज लिख दो. फिर लिखो लिमिटेशंस, ये इसकी लिमिटेशन है इसकी. जब आपका ये चार्ट तैयार हो जाएगा, whenever you are planning to work on any research proposal, so by looking at the chart you can suggest the individual which one is best for your projects. Are you getting my point? Now I am just giving you one task, just for practical, because it's a workshop. So I am just giving you 10-15 minutes. I am keeping on this slide. So you use you take any disease and go through all these databases and pull the data in the Excel file. Pull the data in the Excel file. How many of you want to do that exercise? You do it, then it will give you little bit confidence. Are you getting my point? Swapan, Gatla, Gaurav, what I am saying to all of you is, ये जो आप डाटाबेसेस अपनी स्क्रीन पे देख रहे हो, कोई भी डिजीज ले लो जिस पे आप काम कर रहे हो, इन डाटाबेसेस की वेबसाइट पे जाओ, उस सर्च बॉक्स में उस डिजीज का नाम डालो और टारगेट्स को एक्सेल में ले लो। किसी अगर डाटाबेस से एक्सेल फाइल नहीं आ रही है तो उसको कॉपी करके आप एक्सेल में ले लो। डू यू वांट टू डू दिस एक्सरसाइज? डू दिस एक्सरसाइज। कैन एनीवन पुट इन द चैट बॉक्स? Okay, Gaurav Manoj is saying yes. So I request every one of you do this exercise, and then we will move to the next step. Because unless and until if you are not doing it by your hand, it will not give you the confidence. So I am keeping this slide in front of you. All these databases, क्या करना है आपको? गूगल करो, गूगल में ये डाटाबेसेस के नाम डालो, इनके डाटाबेसेस में जाओ, इनकी सर्च में नाम डालो डिजीज का, और टारगेट्स को पुल आउट करो। ओके, if anyone of you has any query, you can put in the chat box. I will check the chat box. Till now, if you have any queries, you can put in the chat box. Anyone? Okay, so Kavre Manoj, user tech, Tulasida 682, Gaurav, Tanisha, Swapan, Nitesh. Are you doing these steps? Okay, great. You have Gaurav Manoj has uploaded the Excel. Okay, so I am again keeping this means this slide, and within 10 minutes I will see means whether you are facing any problem or not. Okay, do this, right? Continue to do this. ये भी लेके जाना है। I have to. Is my screen is visible? No. Let me share it again. Okay, so how many of you have done this first step, target involvement disease? I think someone of you are facing problem to download in the Excel file, convert it into Excel file, is that. So, why you are facing the problem, as I said, first thing is you read about the database, right? And as I ask every one of you, after this, Gaurav Manoj is saying, no, sir. My voice is audible or not? Screen is visible or not? Can anyone confirm it in the chat box? Gaurav knows is now screen is visible and my voice is clear or not? Or anyone who is online? Is Can anyone confirm is my slides are visible and I am audible? Yeah, okay, great. Sergeant Mori 563 is saying audible. Okay, great. So, what I am telling to all of you, because you all are most of you are beginner, so you will face such kind of problems where you are not finding the option for the Excel file. You don't know how to convert one file into the another file format. So there is an online converter as well. And why you will not get it currently, because as I told to all of you is, ask to all of you is, first read about the database, forget about other things, because to download the target in the Excel file or copy the targets in the Excel file, it's not the hard thing, it's very easy. But first thing is, read about the database, read about their scoring methods, read about their advantages, read about their limitations. So that's why I told to all of you is, आप एक वो चार्ट आते हैं ना बड़े वाले साइज के, वो चार्ट ले लो और ये जितने डाटाबेसेस हैं इनको हर एक कॉलम में डालते जाइए. एंड लेफ्ट हैंड साइड यू कैन राइट, ओके, its source, its scoring methods, its advantage, its limitations, right? Any remarks, any things which you get to know much more about that particular database. Now someone in the chat box has written, sir, you will get the duplicates. Yes, that's why I said, when, see, for example, any one of you is working on yellow fever or anything, so you typed in these database, open target platform, then you have done on dgnet, gene card, omim, hpo, mala card. So how many of you think that by doing same thing in different databases you will get the duplicate? You will get the duplicate or not get the duplicate? How many of you thought that yes, we will get the duplicate? Can anyone put in the chat box? You will get the duplicate or not? Duplicate means, फॉर एग्जाम्पल आपने ओपन टारगेट प्लेटफॉर्म से जो टारगेट है, डीजी नेट के जो टारगेट आए, जीन कार्ड के आए, ओमिंग, कैन दे ओवरलैप विद ईच अदर और दे विल नॉट ओवरलैप विद ईच अदर? Okay, Gauri Manoj is saying we will not get duplicate. Okay, anyone else who want to put their view on this? My question to all of you is, if you are searching same disease in all these different databases, so the target which you will get, whether these target will overlap with each other or not overlap? Guaramano is saying we will not get duplicate. What about others? Okay, and they are providing explanation as well, because every database on different set of database. Okay, different sources of data, right? Guaram knows, not different set of database. Any other who want to answer these questions? Everyone is agree with the Gaware Manoj. You can see in the chat box. Okay. Anyone who is disagree with this statement that we will not get the duplicate? Anyone of you are saying yes, we will get the duplicate? Some will be same. User tech 6sh4wk5h is saying some will be same. Okay. Anyone else? Yes, you will, there is a chances of duplicate. Yes, we will get some duplicate. Yes. Why we will get? Of course, government knows. I think we might get duplicates. Some genes are same because disease is same. Yes, government knows. For example, this open target platform is also providing you data. One of the sources literature. DigiNet is also providing one of the sources literature. This one is also one of the sources literature. So, for example, you got P53. So P53, or even if there is a sources are different also, then also you can get the same target. Maybe P53 is in the literature as well. P53 is in the any other registry as well. P53 is in the particular experimental data. Any data with provide the experimental or clinical trial data. P53 can also be there. So there is a chances of duplicate. Govarem knows. Are you getting my point? Are you getting my point? What I am saying, when you do same thing, even though in different databases, there is chances of duplicate. You just think of, like I have given an example. For example, you type cancer, so in open target platform, P53 can also be there. If you are doing DigiNet, P53 can also be there. If you're doing Malacard, P53 can also be there. Even though their sources may be different as well, then also you can get the same target in different databases. Are you getting my point? Okay, so now question might arise in your mind. Okay, first step we have done, Excel file we have prepared in all from these all databases. Now you have a particularly when you got these different targets from different databases. Next thing is, in Excel there is an option to identify the duplicate. So you put all these targets in one sheet from all these databases, and then identify the duplicate using the Excel, and remove that duplicate. Then after removal of duplicate, your that file will be become comprehensive file for the first step. Are you getting my point? You are saying something, I have query, but if duplicate is there, then why there is not a universal database? Having duplicate means the target. Hari focus is saying what? Having duplicate means target. I think it's not visible to me. Can you put your query again? But Guaremono's query is, if duplicate is there, then why there is not a universal database? Universal database is not easy to create. Universal database means commercial databases are a universal database, which drug discovery companies, pharmaceutical companies are using. They are paying the money to use that data. So these are the comprehensive universal database which will provide you all the information, and provide the segregated information as well based on level of evidence. They will provide this. These are the targets from the experimental study, these are the target from clinical study, these are the target from this source, these are the target from this source. So for these you have to purchase it, their subscriptions you have to be take, then only you can use there. But of course, most of you are in academics, so you are not in drug discovery labs. So there is a limitations, you can't pay much more money because they are charging huge amount of money, like these universal databases, where there is no need to do same thing in a different database. Why we are doing it? Because we are using freely available databases, and every these databases has their own advantages and limitations. So our approach is, we don't want to miss any target. So for example, one target is available in DigiNet, but that target is not available in open target platform. And if you have created your network using only the data of DigiNet, you missed that target, which are available in the other database. Are you getting my point? Okay, can we move ahead? So these steps you have to do. And then, yeah, Pahari focus is saying having duplicate means target is best to study. uh, you can't say that, but of course, means in layman's you can think of, yes, if you have a more number of duplicate, that means that target is more strongly associated with that particular disease. Are you getting my point? Pahari focus. But you can't say that, means it's the best to study. Best to study means there are a certain other criteria as well. Can you please share the mail received from what it? So this is the first steps which you have to do. I hope it's clear to every one of you. क्या करना है आपको फर्स्ट स्टेप्स में? फर्स्ट स्टेप्स में व्हाट यू हैव टू डू इज, आप फाइल बना लीजिए, एक्सेल फाइल्स, put all the targets, and then makes a comprehensive file. I can show you one Excel file, then you will get better idea where it is. 2026. Let me see. Guest lectures 2026. No, I think somewhere I have these Excel files, so I can show you these Excel file. So you will get an idea how to make the Excel files. Some Excel file, right? Yeah, now you can see that. Can you see my screen? I think screen is not visible. Okay. Now can you see my screen? Mm, yeah. Now you can see the Excel file. So you will get an idea how to create Excel files. Now you can see that these are the all the targets. Now can you see the Excel file? So now you can see that these are the genes, these are the databases, right? Nitish saying yes. No sir, not visible. Okay. Now you are saying yes. Yeah, right. So you can see that gene, one column there is a gene. I'm just making it a little bit bigger. So this is the database. So these are the genes which are involved in that particular disease. I forget about the disease, but this is I have pulled in one sheet. These are the targets from the DigiNet in that particular disease. So you can see that these are the targets of the DigiNet. Then you can see I have pasted the targets which I get from the gene card. So you can see that these are the targets from the gene card. Then I have OMIM. From OMIM, these are the targets, right? So like that you have to put all the targets in the Excel file and their sources. Are you getting my point? Now after this, because there is a chances of duplicate, so you have to identify the duplicate. You select it, and in the review or data there is options for the duplicate here. Not in format, conditional formatting. So you just go through the conditional formatting, right? Highlight cell rule. Highlight cell rule, you can see that here is a duplicate values. There is option of duplicate values. Click on it. Now you can see that I got to know that these are the duplicate which are highlighted with the red. So now you can see that these many duplicates are there. So I will remove these targets which are highlighted with red. It means that this target is available in DigiNet as well as in other databases. Maybe it's in OME, maybe in HPO, maybe in Malacard, whatever. And these are unhighlighted one are not duplicate. So what I have to do? I have to remove these duplicates. Then this file will become comprehensive file which contains the targets which are involved in the disease. Are you getting my point? Again I am repeating what you have to do. First step is You have to go through all these databases, put these targets and their sources, put all, means DGNet, HPO, Malacard, put their targets, right? And then of course there is a chances of duplicate, so you have to identify the duplicate. So you just select this, go to the conditional formatting, highlight cell values, duplicate values. So I am putting where is the duplicate, highlight it with the light red, right? Then I click OK. So these are the red ones are the duplicate. So it means that these red the targets which are highlighted with red, they are available in the DGNet along with the other databases as well. So what I have to do, I have to remove these duplicates. So you can remove this duplicate, right? You can put the filter here. Oh, sorry. You can put the filter, and here you can filter by the color. So this is the color, right? So all will come and you can select it and delete it. So your duplicate will be removed. Is it very time consuming process? It's not a time consuming. You select it and remove it. So all these are the duplicate. Got it? You can delete it easily. There is no issue. So this is the first step, and after duplicate remove, this is the file which in file these are the targets which are involved in disease, and these targets are there after removal of the duplicate. Now I have this comprehensive file. Are you getting it? Now again, one step still remain after the databases also. You have to do the literature data mining, right? While literature data mining, for examples, recently in 2 days or 3 days back, new article has been published, and that articles is published in a good reputed journal like Nature, and they have identified the new targets involved in that particular disease for which you are working. So you should not miss any target. So you have to do literature data mining. So literature data mining, I will little bit give you the overview, even though time is less. But you have to put the targets from the literature as well in this sheet, and then after that you have to remove the duplicate. Got it? Any question? That we call text mining approaches. For text mining approaches, we will do separate, hands-on session on how to do NER, named entity recognitions, how to do the text mining, and how to create the knowledge graph. Right? That part we will do, but up to this you understand that this is the first file, and this file will take time, right? Because you have to read about all the databases, their scoring systems, who has created, what are their limitations, what are advantages, and according to research questions you have to pick them. The next step is identification of target of compound, but here only few databases has been added, but apart from that there are other databases as well where you will get targets which your compound can hit. This is the second step in the network. Second step is you have to search, means search the target which your compound can hit, right? So how you can do it? You can do it. Swiss target predictor is there. So these are the already I have explained. This I am skipping this part, but read about these again and again. I am saying, and copy these. Now this is the second step. Second step is identification of target of compound. So compound can be your natural compound, compound can be your newly synthesized new chemical entities, new analog, whatever. So compound can be anything. So you have to be identify the targets of the compound. To identify the target of the compound, again it's very easy process. There is a one of the well known predictor is Swiss target predictor. So just I just show you these, databases are not hard to use, very easy to use. Only thing is you should know about their significance, their score, all these things. So if you talk about Swiss target predictor, just type in a Google Swiss target predictions, right? Click on it, Swiss target prediction, right? You will get like this. Here you can see that Swiss target predictor is providing the results about regarding three species only: Homo sapiens, Mus musculus, Rettus norvegicus. And for example, you are combining your results with experimental part, and you are going to do experiment with zebrafish. So do you think this will be the appropriate database for that particular research work? No, because this database already mentions you can select these three species only. So if you are integrating it with the zebrafish experimental studies, so this is not the appropriate database where you can search the targets which your compound can hit. So if you are planning for after these studies, if you are planning to do in vitro studies on human cell lines, then you can select the Homo sapiens. If your plan is to do experimental studies on mice, you can select Mus musculus. If your plan is to do experimental studies with rat, you can select Rattus norvegicus. Are you getting my point? So for example, I have selected Homo sapiens. So here there is a two options. You can draw the structure of the compound by using this drawing tool, or you can put smiley notation of the compound. So smiley notation of the compounds you can get from that one PubChem as well, Campbell as well. I hope you knows all these databases. From PubChem also you can get smiley notation of the compounds, or you can get it from the like for example any compound you can add. Atenolol I am just putting searching here, right? So there are two options. I can put smiley notations, or I can draw the structure of compound there as well. So there are two options. So I am just copying the smiley notations, but we need conical smiley, so you can click on here. Any questions so far, you can put in the chat box. This is the smiley notations, so I just copy it from here and put in the Swiss target predictor here. If you don't want to draw the structure, so you can see that. But again, when you are copying and pasting the smiley notation of the compounds, just cross check the structure. Sometime if you are doing the mistake in the copy, so if one carbon or anything so you have missed out, so whole process will go in the vain. So cross check it, double sure about it, whatever the smiley notation when I have added, whether this is the structure of atenolol or I have missed something, right? So you have to be careful with that. Then you can click on predict targets. Your job is not working currently, maybe Swiss target predictor, but you have to put the smiley notations, and you can select the means it will take little bit time, not much more. I have just selected other compounds. And sometime also it happens, some database will not provide the targets of that particular compounds for which you want. That what does it mean? It means that database is unable to predict the targets for your compounds. You getting my point? So that's why I am again and again saying, whenever you are using databases, whether it is the databases for the disease or databases for the compounds, make one chart. One chart is for disease databases, another chart for the databases which your compounds can for the compound targets. So you can see that so you can see these are the targets, common name, right? The uniprot IDs, you can see that. And you can download these targets in the Excel file. So you can see there is an option export results. You can put in the CSV file. You click on it, your all targets will comes into the Excel file. So you just open this Excel file. Any questions so far? I cannot find score for Malakad, Klinji. There are scores. You can see that these are the targets. These are their common name. So you have to be pick up the common name. Particularly, Uniprot ID is not required. Campbell is not required, right? But of course, again, you have to read about these 3D by 2D as well, and probabilities. Probability 1 means this is experimental tested and it's strongly associated. So you can see that as the probability value is decreased, that means this particular target, you don't have much more confidence on that particular target. Your compound can hit, but you are not sure about it. So like that, you have to be create similarly the file for the compounds, right? So it's not very hard to use these databases. So you can put it, and again, you have to create files for the different databases for the compound as well. So this is the another databases which you have to use, that is a similarity assemble approach, and it's provided by Department of Pharmaceutical Chemistry at University of California, and it's free of charge. But they have mentions on their database, you use it for your own risk. What does it mean? It means that they are not taking any guarantee about the data confidentiality on this public service website. So whatever the database you are using, freely databases, if you are working on such kind of new molecule, noble molecules, so it's always recommended not to use such kind of databases, because if you are putting these structures, so the confidentiality will not maintain, it will goes. So if you are working on very highly confidential projects and patent is not done yet, so don't use these databases, right? And in this C, that is already mentions, you can use it at your own risk. So this is the, again, C is very easy to use. You just copy the conical smiley notation of the compounds, and this is the C server screenshot. You just put the smiley notation, try C, you will get the structures, right? There is another not structure, targets which your compound can hit. And another database is stitch database, where also you can add the smiley notation of the compounds and click on search, you will get the targets which your compound can hit. Another one is binding db. Binding db, you can write the name in the search box, like you can see that here routine is written, and click on go, you can get the database, you can get the targets which your compound can hit. Another database is CTD, that is particularly with respect to toxicology. Particularly, if you want to understand how environmental exposure contribute to the disease development, you want to understand how heavy metals, how pesticides are leading to disease development, how their genes, their proteins are interacting with each others. You can use the CTD, right? So CTD, here CTD you can very easy to use CTD. But again, before using all the databases, again, read about their scoring methods, read about their sources of data, read about their cutoff values, all these, their advantages, limitations. So you can see that in CTD, which compounds, environmental toxicants like bisphenol, diethylhexyl, phthalate, nitrates, all these. If you are working on such kind of project, air pollutions, pollutants, so don't miss out the CTD, right? This is the comparative toxicogenomic databases. Are you getting my point? This is the CTD when you got the so these targets and their interactions as well. This is for the well certain we have done. And scoring method of the CTD is, it's a quantitative matrix, that is a inference score. An inference score indicate relative degree of mechanistic connectivity between a chemical and disease. Higher score generally reflect a greater number of shared gene and stronger literature supported evidence. So if we talk about cutoff values with respect to that, then inference score above 10, we always consider exploratory. Score above 20 to 30 indicate moderate biological relevance. Value exceeding 50 are frequently interpreted as a strong associations. So this is for the compounds. So create another file, another Excel file, which your compound can hit. So I can show you what kind of files you will get, even though I have put it in the step 1 or step 2. But okay, well certain. This is a B just for hands-on session. This is our molecule, well certain, and you can see that this one side we have added the name of the drug, and another side these are the targets. So before that, you have to put all the target from all the database. These are the targets which well certain can hit, and these are the target which are obtained from Swiss target predictor. Similar way, then these are the targets which I found for the well certains from the CTD. Like that, I have put it all like CTD. Then there is a C database. These are the targets which I got for the well certain from the C database. Then again there is a chances of duplicate, right? So again you have to be selected and go to the conditional formatting, highlight values, duplicate values. You can get to know about how many duplicates are there. These are the duplicates, few duplicates are there, right? These are the duplicates. So remove the duplicate and make the comprehensive file. So these are the after removal of the duplicate, these are the final well certain targets. This is the second step. And of course for this one also you have to do the literature data mining, and you have to add the targets from the literature as well. So after that you will do the removal of duplicate. So now you have a two files. One is file, Excel file, which contains the targets involved in the disease, and another Excel file where you got compounds, the targets which your compound can hit, right? Are you getting my point? Any questions so far? Anyone? So these two files, now these are the foundations. Many time I said these are the foundations file. So as I said, so you have to do the literature data mining approaches to enrich these two files. It's not only the data from the databases, but also do these data mining of the literature. So you can take the data from the Embase, Medline, Cochrane, and so on. But of course, depending upon the availabilities, most of the time, if you don't have access of Embase, even you can use the data from the PubMed and clinical trial websites. So main principle is, with respect to data mining of the literature, is we are converting unstructured data into the structured data. So that is the NER, where we are fundamentals of task of NLP, that is a natural language processing, and it involves identifying, classifying specific entities within the unstructured text. I think this part, these slides I have also shown you. If you, any one of you, have attended the webinar on the graph-based approaches, which we have conducted in the, I think, in last month, and that is available on this YouTube as well. So I am not going in detail, but what I am saying is you have to do these NER exercises as well to enrich your network, right? So your network will be enriched by using these approaches of NER. You can go with the dictionary based, rule based, or machine learning based, right? So but again, to use that, you will first try to use the Python, right? Python or R, so it will be easier to you if you are using this tool. Then you can do it. Then you can go a little bit deeper as well, like deep learning methods, neural network, to understand basic understandings about the deep learning, or neural network. I just add means get this picture from social media platform, where you can see that how neural network recognizing a dog in photo. So this is the dog, right? So you have submitted, so how this dog has been recognized. So before that you have given the training to your machine, where you have learned the machine that this is so this is dog, this is cat, this is rabbit, this thing. So you first make learn, teaching is just like, as I said, teaching a child. So you have neural network sees thousands of labeled animal pictures and learn what makes each other unique. and machine has learned, okay, how rabbits is different from cat, how cat is different from dog, how these two dogs are different, how this one is different from each other. So first you make learn to the machines, and when you have given the input, so it will analyze like this, right? So there is a different layer, first layer, higher layer, top layer, and then you will get the output, right? This is just a simple understanding. When we talk about, you all might heard about this word neural network. So neural network is a part of deep learning. And what is deep learning? What is deep? So deep learning means, you can see that first layer. So network start by noticing basic pattern, as is color, shapes, kind of like how we first notice outline, first texture. Then it goes into deep, higher layer. It combine those pattern to recognize bigger feature, such as legs, कि डॉग्स की लेग्स क्या है, वो दूसरे एनिमल्स की लेग्स में क्या डिफरेंस है, टेल्स किसकी लंबी है किसकी छोटी है, ईयर्स. तो all these, this is the higher layer. Then top layer, here it understanding complex detail, this looks like a dog or not a cat, by matching learned concept from the training, right? And finally you will get the output. Yes, I am 90% sure that, but the picture you have submitted to me, it's a dog. Like, we are talking about these diagnostic, machine learning is playing a role, important role in the diagnosis of the disease, where we are putting all these data of MRIs or CT scans of particular disease. So machine will learn, okay, if this is a pattern, so this is the particular disease the patient have. If I am getting this pattern, so this is disease. So like that, similar way it works, right? Any questions so far? Mhm. So like that, there is a transformer based NER as well, right? But of course, there is a large language model as well, LLM, that perform NER through natural language prompt, often without retraining. So actually, they extract the entities and classifying that, and then find out the relationship between the entities, right? So you are giving raw text prompt instructions, then you are applying large language, then can extract the entities and structured output. So then you can create your knowledge graph or network. So you can get the data from the various sources. That's why I always say, whatever the sources you have, use all the sources of the data and make your files comprehensive. So, and then your network, you can get something from your knowledge graph or from the network. So so as I said, if you are comfortable with Python, but make yourself comfortable with Python, because Pythons are, all these are the need of our, so that's why if you see in recently revised syllabus of pharmacy, also they have included these Pythons. From first semesters, there is a subject has been added, that is the introduction to Python. And beauty of these is Python is freely available. So in Python there is a lot of packages, like you can use size specie libraries, where you can do these NER work. You can create the model as well. If you are very new, then you can go with the pubtatter as well. Pubtatters, from pubtatter you can get the entities, and you can pull these entities in the Excel files, and then you can identify the duplicate and make comprehensive files. And as I said, when we talk about, is my screen is visible? I share it again. Can anyone confirm it? Can anyone confirm it chat in the chat box, that my screen is visible as well as I am audible to all of you? Is okay. Where the Manoj is saying visible, audible as well. Okay, great. So then so if you knows about SLR, so by SLR also you can identify targets which are involved in disease and targets your compound can hit. But SLR should be very comprehensive. If any one of you is doing SLR meta, so you can understand that how much time you have to give for these exercises. So because these are enriching your network, otherwise by creating the network, you will not get much more valuable informations out of it. So you can do these, you can use key terms, fill tag. These we have a separate videos available on this, so you can go through that, how to do these identification of the studies, how to combine the mesh terms with the boolean operator, what is the best way, how to do the search in the PubMed. So all these so session is available on, we have done recently that workshop on SLR meta. That recording is already available, so you can go through that. The purpose is, by doing these exercise, you will get the target and include that target in your Excel file, right? Like if you want to know targets of the routine in the drug resistance epilepsies, so you have to be set up the search strategies where you have to be identify your mesh terms and then combine with the boolean operators, all these. Then there is a AI based tool as well, which will help you to screen it at faster rate. So this is the Elicit, this is the Lit Suggest, Lit Map. Rian is useful for the screening, where you can blind the researcher to each others, and you can do the screening faster. You can include, maybe exclude, and give your comments, so everything will be saved. And pharmaceutical companies are using these kind of tool like digital SR, that is a systematic review softwares. And systematic review, when you are integrating systematic review with the network, your network will become more enriched. That's why I say you learn SLR and meta analysis technique as well. Got it? So that is also required. So covidence is also widely used by the pharmaceuticals companies or CROs who are working with the secondary research. So covidence is a well-known laser AI. Now next step is, when you've done all these steps, and these two steps, whatever the steps I have explained to all of you, these two steps are very important with respect to network. Give your lot of time to these two steps, and most of the times students or researchers who are working or using these network techniques, they are doing the mistakes in these two steps. And if you are doing mistakes or anything, not making it comprehensive, not giving time for these two steps, later on whatever you will create, analyze, that is based on these two steps only. So that's why you perform SLR as well, and in a proper way include that targets as well. Then identify the duplicate, then remove the duplicate and make the comprehensive file. Anyone have any questions so far? So these are the two steps. Now we are moving to third step. But before that, if any questions, any discussions you want to do, anything else? Okay, if there is no questions, so we can move ahead. So next step is, now third step is, you have to identify the common targets, right? So you can see that this is the first steps. Excel file, I have a well certain target, right? This is the uh, another steps, where I have disease targets involved in the disease. Now I want to get to know about what are the common targets. That is the step 3. So to do that, you can do vanning. Vanning is very easy to use, where you can identify the common targets. So I am doing vanning. Vanning, you just type vanning, right? Vanny 2, 2.1. Okay. See, this is the Vanny 2.1, right? Now I want to identify the common target. So what I will do? I will do the targets which are involved in disease and targets which my compound can hit. I will copy these targets after the removal of the duplicates. So I copy these targets. There are so many. I have just added few. You can add these target in one side and another target in another side, right? My second file is, let's see how many duplicate, how many common will be there, because I have just selected few only. Now you can, there are so many, right? I have selected it. I put it in the Veni in the other side. Now you can see on your screen these 29s are the common. These are the targets. One set is representing the targets which are involved in the disease. I have copied it from the Excel file which I have prepared after the removal of the duplicate. And one side I have added the targets which my compound can hit after the removal of duplicate. Got it? Then when you have added to both sides, you will get this common. So common is 29. So I have just clicked on it, and these are the 29 targets which are common. That means these are the targets which my compounds can hit. But these are targets are not in isolated form, so I copy these targets. So next step is I want to check the interactions among these targets. So for the interactions among the targets, the powerful tool is string database. So just type in a Google string database, click on it, click on search, select multiple protein options, paste here the common targets which you get after doing the Venn. And now you can select in species as well, or if you want to take it auto detected, it you can do it, and then click on the search. So by the string you will get to know that how these targets are interacting with each other. Got it? So for example, I have selected Homo sapiens, click on search, and then click continue. Now you can see that. What are these circles? Can anyone type in a chat box? Now you can see that interactions among the common targets. So what these circle represent? Anyone in the chat box? What these circles represent? Can anyone put in the chat box? Anyone can put in the chat box. Okay, just 1 second. Hello. हाँ, बोलो. घर पे हूँ, बोलो. क्यों? क्या बोल रहे हो? हम्म, हम्म, हम्म. मैं अभी देखता हूँ. देखते हैं, मैं 2-2:30 बजे के बाद आऊँगा. हम्म, देखता हूँ अभी मैं. Anyone can put in the chat box what you understand. Okay, notes. Okay, great. Zagarg is saying nodes, proteins, node. Yeah, circles are node, and node can be your, your proteins, your genes. And what are these lines which are connecting these nodes? And if you see, there are a different colors. Can you see the colors? The colors are different. Some are red, some are blue, right? So what these color represent? You can see below these colored node. What are these colored node? What are empty nodes? What are filled node? Are you getting my point? You can see the colors. Yeah, relationship, it tell us about the relationship. So you can see that when you are explaining these particular network of the interactions of the among common targets, right? So you can explain about about these color, how they are interacting, and you can export these figures from here as well. You can export it as a download as a image or like that, right? Any question on this? So you can read about color coding as well. Earlier we are not getting color coding, but now string or these are providing the color coding as well. Any questions so far? Just give me 1 second. I have to be forward one email. Meantime, you can put your queries in the chat box. Anything. So what you understood so far? How many steps you have done? Right. One step is okay. So what I am saying is these are the networks, and these you have to read it. You have to see analysis of network is very important, because I have just added a few of the targets, but so many times there is a lot of connections, lot of targets. Even though in this network, can you can you validate experimentally all the targets? It's not possible. So you have to be select which are the druggable targets, which targets your compounds can hit in that particular disease pathogenesis. So that we will discuss in the analysis of the network. So up to this, you understands that how to identify the common targets by the venning, right? So you can see that simple exercise. You can, one side you can add the your compound target, another side you can put the disease targets, right? And you will get the common. Then interactions among the common targets you can check by doing the string. Just type in a Google string, you can check the interactions. So string also you have to read about the string. What is a string? What is cutoff value? So cutoff value is more than 0.7, and setting the cutoff lower will increase coverage, but if you are decreasing the cutoff values, it will, of course, you will increase the coverage, but it increase the fraction of false positive. So this is a string database, very popular. In every network paper you will find this string database. So here you can add the common targets, and then you can select the species, and you can check the interactions among the common targets, how common targets are interacting with each other. There is another one is BioGrid. BioGrid is also the database of protein, genetic, and chemical interactions. Here also you can check the interactions among the databases. Another one is DIP. DIP is the database of interacting proteins that also can be used And intact is also popular one, that is a molecular interaction database, how targets are interacting with each other. So you will get the informations by using the intact. Then another thing is, so when you are creating any network, or so you are analyzing their interactions, so I always say you can go with the R package. R is the very powerful tool, not only for the network, for all other analysis as well. So install R in your laptop or in your computer systems. So these are the site where you can install the R, and you can run it on window. So download R for the window, install R if you are doing it first time, and then with R install the R studio. So in R studios it will be open like this, where you can put your commands. You can first step is, you have to change the directory, then you can install that particular package, and then you can unpack it, and then you can import the data, whatever the data means, whatever the data you have collected, you have to import it into it. It will create, and igraph is the one of the package for the R. And then you can create the network and analyze the network. So this is the just a screenshot of the network which is created by the R, right? And this is a Python also you can use it. Pythons, the module is network X, which you can use for the network creations and analysis of the network. So you can go with the R or Python, it depends upon you, whatever you are comfortable. But by using these, and both are freely available, by using these you can do lot of analysis of the network, and you can create these so you can understand the parameters. So now we are getting into the most probably the last part, that is the analysis of the networks, right? How to analyze, you have created the network, how to analyze this network. So to do the analysis, as I said, first you have to understand about the basic parameters, what are the parameters, and what that particular parameter tell us about, and which one is important. There are number of nodes are there, number of proteins, number of connections. So as I told yesterday as well, the proteins which have lot of connections, that means that protein might have a important role in biological function as well. So you can't pick up that target for the drug discovery and development, right? So analysis of the network, first you have to understand how you can express network into the mathematical form, in the form of urgency matrix. And by using the matrix you can calculate parameters, network properties such as degree, another centrality measure, by applying basic concept from linear algebra, right? So this is the how to convert network into the urgency matrix. Network can be of many types, graph can be of many types, we have discussed yesterday as well. It can be directed, it can be undirected, it can be weighted. So you can see that if I want to convert this undirected graph into the urgency matrix, so first is we have to set the dimension of urgency matrix. So how many dimensions should be there? So dimensions of the matrix we are deciding based on how many nodes are there. So you can see that 1 2 3 4 5 6 7, so 7 nodes are there. So my urgency matrix will be 7 by 7. So I have written A B C D E F G, A B C D E F G. So now A and A, so A is connecting with itself, no. If there is a arrow A to A, then I will give the 1 marks. A I J, I is the one node, J is another node. If there is a connections, I am giving 1. If there is a no connections, then I am providing 0. So you can see that A is not connecting with itself, A, I am giving 0. Then A, whether A is connected to B, can anyone answer in the chat box? A is connected, A node in the first graph, whether A is connected to B node, yes or no? A is in this graph, A is connected to B node. Yes. Yeah, if it's connected with B node, I have to put 1, right? So now you can see that A in front of B, I have put it 1, right? Yeah, you are right. Then whether A is connected to C node, A to C, yes, right, 1. A is connected to D, yes, 1. A to E, yes, 1. A to F, 1. A to G, 0. I am giving because A is not connected to G, right? Then goes to the second node, B node. B is connected to A, yes. B is connected to B, no. B is connected to C, so you can see that 0, 0, 0, 0, 1. 1 is giving because B is connected with F, right? B is connected with the F, so I am giving 1. So like that, when you convert your network into the adjacency matrix, you do the summations, you will find that this A node, in this particular network, this A node has a 5 degree. 2, 1, 1, 3, 1. So in this network, which node has the highest degree? Which node has highest degree in first graph? Which node has, my question is, which node has higher degree in the first graph? And degree means yeah, A, you are right. Degree means how many connections particular node has. So in this network, because this is a simple network, so by looking at the network you can calculate, which one has the high degree. So A has the high connections, right? One, 2, 3, 4, 5. 5 connection A has, whereas as compared to other nodes, they have a less than 5. So when there is a biological network, big network, so computers are doing these calculation, and they are providing the degree values to you. So it's a simple network, but you know that biological network are not the sparse. Biological networks are the dense network. So in dense network, manually it's not possible to calculate degree or other parameters. So computers are calculating it, but I am providing you basis how computer is doing the calculations. Are you getting my point? So how computer is doing these calculations? So they are converting it in the adjacency matrix, and by the adjacency matrix, you can calculate the parameters. Similarly, you can see that second graph is your directed graph, and another one is your what kind of graph this one is? This is a weighted, but whether it is directed or undirected. Third one, can anyone put in the chat box? Third graph is which type? Whether it is directed or undirected? Of course, weighted is written because weightage is given, but what about uh, directed or undirected? Whether is it directed or undirected? Third one. Can anyone put in the chat box? Anyone? Third graph is, of course, it's weighted. Yeah, undirected. Subham is saying undirected. Manoj is saying undirected. Yes, it's undirected. But all graphs you can put in the, uh, you know, in the urgency matrix, and you can calculate, right? Here in the, when there is a weightage is given, you are giving, because weightage is given for the connection of the node, so you are giving the weightage marks, right? Instead of 1 or 0. 1 or 0 we are considering because there is no weightage between the node, so we are giving standard. Okay, if there is a connections, I am going to give 1, whether the connection is between this node to this node or whatever it is, I am giving, if there is a connection, 1. If there is no connection, I am giving 0, right? But when weightage is already provided, so I know, okay, this connection is stronger, this connection is weaker, this connection is stronger, so I have to be convert into the urgency matrix according to that, right? Means like you can see that A to A is 0, but A to B is 8. So you can see that A to B connections, the weightage between A to B is written 8. So that's why I am writing here 8. Getting my point? So degree, as I said, degree is very easy to calculate. We are talking about number of edges that connected to a node. So if we talk about the simple network, these are the node, these are the vertices, these are the line or edges. So if I calculate the degree of A is 2, because what is degree? Degree is the number of edges connected to a node. So how many edges connected to the A node? 2, right? B 2, C 2, D. What is the degree of the D node? Can anyone type in the chat box? What is the degree of D node in this? Can anyone put in the chat box? What is the degree of D node in this? 3, right? Because degree is a simple measure which tell us about how many connections particular node has. So you can see that degree, if you want to calculate the average degree of this network, what you have to do? You can formula is 2L, or you can call it as is divided by total number of nodes, right? So 2L edges are how many edges? 5. So 2 multiplied 5 divided by 5, 10 by 5, it's 2, right? So degree is 10. If you want to calculate average degree, 10 by 5, it's a 2, right? So you can do these calculations. But sir, depending upon the graph, because it's an undirected graph, if your graph is directed, then there can be a two types of degree. It can be in degree or out degree. So when there is a graph is directed one, so directed one means there is a direction is given. So you can see that for a node how many in degree is 1 2 3 4, and out degree is 1, right? Can anyone tell me about but about F node? How many in degree and how many out degree of the F node? That is the directed graph. F node. You can put in the chat box, even though it's visible, but of course you can check. What will be the in degree and out degree of F node? So when we are talking about a directed graph, there are two types of degree: in degree and out degree. Because direction can be towards the node, direction can be away from the node. There are two possibilities. So if the direction is towards the node, we call it in degree. If the direction is away from that particular node, we call it out degree, right? वो उसके बाहर की तरफ जा रहा है एरो, और जो अंदर की तरफ आ रहा है उसकी तरफ आ रहा है, that we call in degree. That's simple. So you can calculate in degree and out degree of each node, right? So my question to all of you is, what will be the in degree and out degree of F node? And anyone put in the chat box? Out degree 2, in degree 0. Out degree 2, or you are talking about F node? Yeah, F is 3, right? Out degree is 3 and in degree is 0, right? Pahari focus is saying yes. Okay. 0 in degree and 3 out degree, right? This user tag is saying, right? Yeah, right. So depending upon the types of graph, you have to be report the parameters. So when your computer is giving the values, so you should understand each and every parameters. So now your basic concepts in your mind, knows about, okay, if we are talking about directed graph, there should be the in degree and out degree. If someone is presenting only the degree, then question will arise. If someone is knows these basic concept, they will ask you, for directed network, your network is directed, why you are presenting only the overall degree, why not in degree and out degree? How much in degree and out degree is there, right? So then you can plot degree distribution as well, and by cytoscape or these kind of tools, you will get these kind of graphs as well, by igraph as well, so this one, Python or R package also, you can create such kind of graphs. What is the purpose of this graph? This graph will tell you about, in particular biological network, which node has higher degree, how many nodes have, for example, 5 degree, how many nodes are there in the network which has 5 degree, how many nodes in the network which has highest degree, how many nodes in the network which have a less degree. So by the degree distributions you will get to know about that, right, in the biological network. So according to this degree distributions, you can write your write up. Okay, as shown in figure 1, this degree distribution, these many nodes having the high degree, these many nodes have a lowest degree, these many nodes have more connections, all these things, right? Then next parameter is density. Density is the, when you calculate the density tell us about fraction of all the edges in the network. So the formula for this fraction of all the edges in the network, if your graph is undirected, that is a 2 multiplied by L divided by n minus 1. For directed, it's a L divided by n minus 1. Now my question to all of you is, why there is a difference? What is the difference in these two formulas for directed and undirected? That is my first question. Is there any differences in their formulas when we calculating the density? Density is the fraction. You are calculating the fraction of all edges in the network, right? Can anyone tell me what is the difference between these two formulas? First tell me the difference and then tell me why there is a difference. Anyone? Are you getting my questions? What I am asking? My question is, on the screen, you can see there are two formulas for the density. And what is the density? If you are getting density values, so density is the fraction of all the edges in the network. So if you look at the formulas, first tell me what is the difference between these two formulas and why there is a difference. Anyone? Yes, Gaware Manoj, Pahari Focus, User Tag, Subham, Isa Garg, and Thula Asidas, User Tech 6SH, Isa Garg, Sanjan Maurya 563, Pahari Focus, Nitish Kumar, Sanjay Mauri, Gaviraj Manoj, Sanjay Mauri, Nitish, Pahari Focus. Undirected have double the value of the directed. Okay. Thula Asidas 682 is saying undirected have double the value of directed. Directed is in particular path. Okay. Anyone else? Anyone who want to add much more? Yes, anyone else who want to answer these questions? You are close to answer. Yes, you are right. This may be because we don't what they are connected. Yeah, means undirected, as I told you, if you remember yesterday as well. Undirected means it can go in the both the sides. It can go A to B, B to A as well. That's why two. There we are multiplying with the two links, right? One node can have a two links. So how two links? Because if there is a and B node, so A can go from A to B and B to A as well. That's why in undirected we are multiplying with the two, because it has a two links, whereas directed there is a particular directions, right? So this is a density. That is another parameters which you have to be get the value by using these Python or R package. That is a clustering coefficient. So what is a clustering coefficient? What it tell us about? Clustering coefficients measure the connection of the neighboring node to each other, right? Neighboring nodes. So now my question to all of you is, for example, this is the A node in this square box, B node, C node, D node. So what will be the clustering coefficient of A node? Can anyone answer it? First, understand what is clustering coefficient. Clustering coefficient is measure of connection of the neighbors. Again, stress on connection of the neighbors node to each other, right? So connections node to connection of neighbor node to each other. So what will be the clustering coefficient of A node if we talk about this second one, this square box, A node? Tell me in the chat box. And sir, you can also one another thing is, you all can also put your correct name in the chat box as well, for the certificate. If we create the certificate, one, Subham is saying one. For a node, clustering coefficient is one. Okay. Any other one? Or you all are agree with the Subham that a node? I am again talking about this square box. This square box, and this is the a node, this is a b node, this is a c node, this is a d node. Okay, Guare Manoj is saying clustering coefficient of a node is 2. Okay. Any other? Tula Tula Didas user tag is saying 2. Okay. Pahari Focus is saying what? Or any other? Isagar, Gaurav Kisorkar, what's your point of view? Sanjay, Guare Manoj, Sanjay Maurya, what do you think? Nitesh Kumar, Isagar is saying 2. So everyone is saying 2, but it's 0. It's not 2, it's not 1, it's 0. Why? Now question is why I am saying 0. See this A. First read this clustering coefficient definition again. Clustering coefficient is a measure of connection of neighbors. Connections of neighbor node. So first you just think of, this is the A node. To A node, which one are the neighbors? Tell me about that one in the chat box. A के नेबर कौन है? इसमें ए के जो पड़ोसी हैं वो कौन कौन है? कौन से नोड है? टाइप इन चैट बॉक्स इमीडिएटली. ए नोड के पड़ोसी कौन है? डी एंड बी, राइट? राइट शुभम, डी एंड बी आर द नेबर्स टू ए नोड. नाउ whether D and B are connected to each other, whether D and B are connected to each other, yes or no? No. So what will be the clustering coefficient? Not connected. So what will be the clustering coefficient? 0. So clustering coefficient will be 0, because what is clustering coefficient? Clustering coefficient is how your neighbor nodes are connecting to each other, and the neighbors to A node is B and D, and B and D are not connected to each other. Are you getting my point? You, someone of you are saying 1 or 2. Got it? So that basic. Similarly, what will be the clustering coefficient of B node? Now you can answer. B node. What is the clustering coefficient of B node? Avdesh Infotech is saying undirected. 0, right? Clustering coefficient of C 0, because B node neighbors are A and C, right? C node neighbors are B and D. D node neighbors are A and C, and they are not connected with each other. That's why their clustering coefficient is 0. Now in these pictures, you can see that which network will have a high clustering coefficient. This one or this one? This one will contains the high clustering coefficients, because if we talk about this a green node, and what is the clustering coefficient of green node, and here what is the clustering coefficient of green node, so of course high clustering coefficient will be this one, right? So I hope you got clustering coefficient concept. Then another parameter you oftenly heard when we talk about network is shortest path and diameter. Shortest path. So shortest path, simple understanding, shortest distance between any two node. Shortest path means what? Shortest path means shortest distance between any two node. So for examples, if you want to go from this node to this node, this node to this node, for example this one node and this node, you want to go from one node to this node, right? So there are a many paths by which you can reach up to this. So you can go from this to this, then this to this, then this to this, then here, then here, then here, then you can make it circle, then you reach back. So we are not considering all the path when we are calculating. The shortest path means the shortest distance between any two nodes. So if we talk about, this is a starting node, this is the end node. So I want to go from this node to this node. So which one is the shortest path? I can go by this way as well, and I have a multiple options. Just like you are going somewhere, and your Google Map is showing always the shortest path, right? So like to reach that particular destination, you have a multiple path. So you want to see which one is the shortest path. Similarly, from one node to another, which one is the shortest path. So that is the shortest distance between any two nodes. That is the shortest path. If someone asks you, okay, what is the shortest path with respect to network biology, tell me about that. And what is the diameter? Diameter is the length of the shortest path. उस shortest path की जो length है, that is for example, you are going from here to another place, and one path is going 20 kilometer, another path is saying 14 kilometer. So that is the distance, 14 kilometers. The length of the shortest path we call diameter. Got it? Then there is a centrality measures. So centrality measures is very important parameters when we talk about network biology. And centrality measure tell us about which node is important, because there are so many nodes, so many things, so many connections. Which one is important, right? Sometimes the node with the high connections is not that much important, right? Of this is saying one in degree, zero out degree. I think of this. Okay, that we already discussed. Node is connected with at least two nodes. So centrality measure is degree centrality. This is the normalized value of degree, and there is a in degree and out degree centrality when we talk about directed one. Then there is a closeness centrality. Then there is a eigenvector centrality, betweenness centrality. So you can see that this second node in second graph, degree we have already discussed. So degree is the this are normalized values of the degrees. But betweenness centrality, you can see that the second node, how much degree, what is the degree of second node in the second graph? Can anyone answer it in the chat box? सेकंड ग्राफ में सेकंड नोड जो रेड से हाईलाइट है, लिखा हुआ है इस पे तो इसकी डिग्री कितनी है? व्हाट इज द डिग्री ऑफ दिस? क्या डिग्री है उसकी? 2. इस अगर ये सेइंग 2, राइट? Two. Tanisha is also saying 2. You are also right. So but other node in this network have so many degree, like this one, 1 2 3 4 5 6 degree, right? Here you go to another network, 1 2 3, here 1 2 3 4 5 6 7, like that, right? So now between a centrality, even though this node, second node, has 2 degree only, but maybe this node is important for me. Why? Because this is a connection between the two things, two hubs. This is a connection between the two hub, right? And if I am able to break these connections or inhibit these connections, disease progression is going to reduce significantly. So in that case, which node is important for me? This second node, even though this node has low degree as compared to other nodes in the network. That is the betweenness centrality. Are you getting my point? So that's why when we are doing the analysis, we are looking for the network, we are looking for all the parameters, then we decide. See, do you think is it a easy task to identify a new targets? It's not a easy task. You have to be. Otherwise, what will happen? You people are doing, you are doing these exercises, and finally you are getting only those targets which are already known, and expert will say okay क्यों किया ये सब, ये तो पता है ना, इसका तो मुझे पता है कि ये टारगेट इस पे तो ये चल ही रहा है, तुमने ये सारी एक्सरसाइज करके निकाला क्या, right? So that's why when you are doing the analysis of the network, you have to be very careful. You have to understand all the aspects. So betweenness centrality is important for such kind of networks. Closeness centrality also you have to calculate how close these nodes to each others, one node to another node. Eigenvector centrality is also another measure. So what is eigenvectors centrality? Eigenvector centralities measure the importance of a node based on its connection to the other important node. So to make it simple, you just understand you have a two friends. One friend has a 50 connections, another friend have only 10 connections, 10 friends, right? Another one has a 50 friends. But that 50 friends, none of them is important. मीन्स काम आने पे वो उन 50 के 50 कोई काम नहीं आएगा आपके, और जो 10 हैं उनमें से आपके 8 काम आने वाले हैं, तो विच वन इज मोर इम्पोर्टेंट? सो यू कांट से दैट कि उसके 50 फ्रेंड हैं तो उसका ज़्यादा इम्पोर्टेंस है, राइट? वो काम के तो है ही नहीं. तो इगन वेक्टर सेंट्रलिटी कि हाउ मच इम्पोर्टेंस ऑफ नोड बेस्ड ऑन इट्स कनेक्शन टू अदर इम्पोर्टेंट नोड, कि उसके कितने इम्पोर्टेंट कनेक्शंस हैं आगे, राइट? एक फ्रेंड है उसका कनेक्शन सीएम के साथ है, तो वो आपका फटाफट काम करवा देगा, भले ही वो दो ही हैं लोग, दो ही फ्रेंड हैं, पर दोनों मतलब उनमें से एक सीधा आपका सीएम के थ्रू काम करवा देगा या पीएम के थ्रू करवा देगा, और बाकी है वो उतनी इंपॉर्टेंस उनके कनेक्शन है ही नहीं, भले ही वो 100 है या 200 है, बट दे आर नॉट इम्पॉर्टेंट. हेयर वी आर टॉकिंग अबाउट इम्पॉर्टेंट फॉर development and progression of the disease. Got it? So that we are calculating by calculating the eigenvector centrality. Important node, less important node, right? Any question on this? Okay. So there are a, this is the one of the very important when we are selecting the node, druggable node. So there are a central hit strategy is there, network influence strategy is there, adjective drug strategy is there. So again, that we will decide once you will create the network, analyze the network, all the values you have, then we can go with either of the strategies to identify the drug target. Right. These are the recent works which we have done in our labs with respect to network, these data mining approaches, where we have yesterday someone is asking about the signals, so we have done the signal analysis, then we have coupled it with the network, then we have done the molecular docking, then dynamics, then we have done the experimental animal studies for the validations. So this is a complete set of the work where we have identified the repurposing molecules for the hepatic encephalopathy using pharmacovigilance based approaches integrated with experimental verifications. Right? These are the pathways which we have identified, and these are the drugs, repurposed drugs, amlodipine and carvedilol, where they can hit. We have also applied these strategies for the natural compounds, how natural compound can be useful against drug resistance epilepsies, right? So we have done again same process we have done, we are doing coupling it with the molecular docking, and I have told you yesterday as well, docking will tell you about energetically favored binding conformation in the active site of ligand, but network will only tell you about possible targets or possible pathways. But how your drug is interacting in the active site, you will get to know by the docking, and how much stable it is in the active site when you are doing the dynamics, so you will get by the dynamics, and then you can couple it with the experimental studies as well. So this is another one we have done the experimental means same flow, where we have done network, then molecular docking, then dynamics, then experimental animal studies. This is another work where we have explore the selected phytoconstituent as a galactose using network and experimental approaches. We have done these studies in the rats. So all these parameters we have calculated, then selected the targets for the docking, and then dynamics, and then experimental. This is another work where we have coupled it with the Tinospora cordifolia, how it enhances the immune response in the mice immunized with the JEV vaccines, where we have applied the network pharmacological approaches with the experimental approaches, and it's published in Phytomedicine Journal. So these kind of works we are doing, these novel signals, then network, then docking, and these are published. These are the recent research studies where we have used these networks, molecular docking, dynamics, and preclinical evidence evaluation as well. These are the recent studies which are published in International Journal of Repute with respect to networks, data minings, and these one. This is our ongoing studies. We are working on understanding the biological network of MASLDs under ANRF pair projects, where we are currently working by using the Python. And this is the book which I have edited with Professor Patil on network pharmacology, principle, processes, and applications. And these books cover from basic to advanced levels. We have included basic chapters initially, and then we have added the case studies as well. So in case studies, we have step by step. If someone want to do apply the network pharmacology, how that researcher can apply these techniques. This book is available online as well, right? Or you can recommend to your library as well. Then next is challenges with respect to network is data is very important. So that's why many times I am saying to all of you is, whatever the database data you are using, just read about the database. Otherwise, if data is not of standard qualities, or data is not important according to your research questions, so it will create a lot of noise in your networks, right? So you will get difficulty when you are doing the analysis of the parameters. You will not understand anything out of it. So that's why you have to be checked. Quality of data is important. Multiple data sources are there. So these are the current challenges. An international standard for network pharmacology is going to come soon, because currently, otherwise, we are everyone is using their own steps, but that is not the right way. There should be the standard methods by which you can make your network comprehensive, and you can give much more times. So conclusion is, it's a next generation medicine research mode. More in-depth analysis is required, and rapid evolving field. So you have to be update yourself. As I said, there is a lot of data is being generated. So that we are talking about big data analytics. A lot of data is there, but problem is to analyze that data. So analysis is very important, plays significant role, and in-depth analysis is required. And whenever you are doing the analysis, you try to compare it with the these particularly clinical data as well. This is all about. Anything else? Any questions? I am happy to take your. I'm going to stop here. Anything else? Anyone can put in the chat box. Anyone? I hope every one of you got an idea. If you are very new, of course, so slowly and slowly you will learn it in much more detail if you find it interesting. But this is just a workshop for for beginners only. So I assume you all are beginners. You have not used these techniques earlier, or if you have used, not used comprehensively. My camera is not working. Let me see. Any questions? Anything? So I hope with these 2 days you learn many things, or you got to know about right, what are the different databases, how to pull the informations, and how to make the Excel files, right? Any questions, anything? I can see this one, mobile phone as well. If you have any questions, you can put in the chat box. Sir, our drug is not plant based, then how to get their phytoconstituents? If your drug is not plant based, if it's a synthetic, so you draw the structure of synthetic molecules. We are applying the network techniques for the synthetic as well. There is no issue with that. See, it's not like that drugs should be herbal only. You can apply these techniques for any molecules. It can be from marine sources, it can be synthetic, it can be anything, whatever, right? Next question is, multiple of two node as connected with at least two node, not at all. The how to examine KEGG pathway and geo enrichment. KEGG pathway, you can do the SR plot analysis. So you just type in a Google SR plot. So after creations of the network, by so by doing these exercises, you will get to know about the targets, how these targets are making that particular pathways. You can do the SR plot. So if you type in a Google SR plot, you put the all these targets, it will create the pathways. What are the possible pathways that can be done? Geo enrichment. If our drug is not, so I think Tanisha, is it okay? So maybe you are working with synthetic molecule or something. Next is how to plot graph of fraction of nodes versus degree. You can do it by the Python or by the R package as well. So that's why I am saying to all of you is use these. Maybe initially you will feel uncomfortable when you are using Python or R package, but I always say please use slowly and slowly you will become comfortable. And then why I am telling this, because by using these Python or R, uh, it's not only for the network. You can do lot of other analysis as well and free of cost. you know when we talk about meta analysis, in meta analysis workshop also we say that use R, use Pythons, particularly when we talk about disproportionality analysis, when we are doing with respect to pharmacovigilance databases. So in that one also use these. So what I mean to say, by using these particular Pythons or R, you can do a lot of statistical analysis, lot of these GWS studies, gene wide association study. If someone of you want to do gene wide association studies, so by using R and by using Python, you can do the gene wide association studies. If we talk about NER, named entity recognitions, if you want to do that, if you want to apply the large language models, if you want to do machine learning work, Python and R, right? So that's why I not suggesting Cytoscape many times. Of course, Cytoscape is easy to use, window based. You can import and it will create beautiful networks. But use these Python and the R package. And as I said, we I will share videos as well, how to use these Python, how to install it, how to use it for the network. I will share that part as well, links of that as well. Okay. Anything else? Any question? Okay. So if there is no questions, okay, how to use R and I graph? Yeah, Guarem knows. I will put the link in the chat box. Okay, Subham, thank you for joining. I'm really grateful that you organized this workshop. So I hope means from this workshop, many of you will implement these techniques in your research work, and you will make your profile stronger, and you will be reach at the heights in your professions. And of course, when you knows the standard things, of course hard work is required, but it will give the confidence से आपने किया है, so it will be reflected when you are presenting to someone else, so it will be sense. that okay, मतलब ऐसे ही है. When you have a means it reflected in a different way. When you have a good knowledge, strong knowledge, expert in that particular whatever you have picked up, it will give you the different set of confidence. Okay, user tech is saying thank you sir, it was very informative workshop, nicely explained why in a simple way. Thank you very very much. Thank you user tech 6. Thank you for joining. Isa girl, thank you for this workshop. Isa, thank you for joining. Tulla Asida 682, thank you sir. Thank you everyone. Anyone, anything else? Sir, in SR plot we have to add date from when. No, not date. SR plot, you can add the target. Means your compound can hit in that particular disease. So I will try to make that video as well and put in the channel as well. How to because that I think that part is still left. Pathway enrichment analysis. So that I will explain to you in that separate one. Right. Thank you so much. Avdesh is saying thank you, Avdesh. Thank you, Gary Manoj. So thank you every one of you for your time. So enjoy your learning, and if you have any things come out when you are doing it, you can put in the chat box. Right, this video will be remain in the YouTube. So in their chat box you can put your queries. Sometime maybe if I am not reverting back, just have a patience. Whenever I get time, I will reply. That's why I am little bit, I can give the much more time. But once the sessions will be started, so it's sometimes it's very difficult to look for all the queries and respond to it. But I will try my best. You keep putting